Sir, you're live now. You can start.
All right. Good morning, everybody. Welcome to today's live session. Today, we are going to plan to give you some demonstrations from the lab, along with interacting with you live. So this could be your opportunity to talk to us and ask questions. But while you are going to do that, we are going to show you some experiment in a more thematic way. Today's theme is going to be on integrating multiomics data set to understand the different biological questions. And in this particular session, we are going to show you some live experiments from different type of proteomics and genomic technologies. But then more focus will be how to do the data analysis and integrate data from proteomics and genomics. So rather than making it very theoretical, I thought to keep it very minimal in terms of talking to you on the slides, but we do more hands-on in the lab. But I'll give you a very quick flavor of where proteomics has started making more impact to understand biology and addressing many clinical questions, right? So something which is coming up very new is about single cell proteomics. And I thought to take this as the first topic because one of the students, Srijita, she asked a question about how the field of single cell genomics and proteomics is emerging or going to really make impact. So in this slide, I'd like to say that single cell genomics, mainly single cell transcriptomics, is already very established and successful approach. And people realize that they can do sufficient RNA and do the transcriptomic analysis with RNA-seq. But the challenge was to look at from single cell how to do the entire proteome analysis. But now with the advancements of the new type of mass spec workflows and the new sample prep, it has really now started becoming a reality that one could even look at the proteome from single cell, right? So what I showed you here on the slide, think about if we have to work on the cancer, to look for the unique proteins linked to a given cancer. What we do in our own research, which is a reality, and others also do, we take the tumor tissue, and that tumor tissue is a lump of the mass of many type of cell, right? We homogenize them, lyse them, and in a hope that we are looking at only cancer related protein, but that's actually not. No, that's not true because we are mixing a variety of cell type. In this example, if you see neutrophil, macrophages, blood vessels, T cell, fibroblast, all of them along with the cancer cell. Now, if you were to disaggregate this tumor mass and look at separate components of them, then only you can see you have very few cancer cells, but there are a lot of other type of abundant cells which are present there. So in which way we can try to first look at the cell sorting and look at a single cell of our interest, which we are keen to look at the proteome or transcriptome. And based on that, now, can we do sufficient ways of sample prep and enrichment that we can get a lot of peptides from that given cell, and one could run in the mass spec what you see in the bottom panel to generate those mass spectrum data and then look at the data analysis and come up with a lot of different type of patterns. So this was definitely not possible a few years back, but now from last year and so, there has been a lot more progress being made in this area that now we have good ability to look at the from single cell even close to 5,000 proteins, 3,500 to 5,000 protein is what people are able to now achieve with the newer advancements of the mass spec like timsTOF and Astral Orbitrap. One could really go to much deeper coverage with the single cell as well. But also like the sample prep becomes very crucial because you need to make much more automated processes so that you're not losing the much peptide. And also you are looking at how best to preserve the peptide which you lost in the multiple steps. So single pot analysis, scopes method. There are many type of new workflows which has emerged, which I'm not going to talk as a lecture here, but that just to give you flavor that single cell genomics and proteomics and soon single cell proteogenomics will be a reality, which is almost, we are making good advancement in this area. So now just another exciting thing which is happening in the proteomics right now to look at the cell surface proteome. While most of the time we take the entire cell and try to lyse the cell and look at every protein inside that, which is the global proteome. But if you think about a lot of drug targets, they work only on the surface, right? So we should be focusing for the drug target discovery more on the membrane protein and the things which are more receptors and present outside the membrane. But when you combine everything, you are actually diluting with the global abundant, highly abundant protein to these low abundant and more unique targets. So how to enrich only the specific cell surface protein is another emerging area in the proteomics, which are a couple of good successful examples shown here with also our collaborator at UCSF who are developing the new workflows to only enrich the cell surface protein. And again, with these advanced mass specs, it's possible to analyze thousands of protein from these type of unique cell surface protein. But again, biologically, these are very important development which are happening in the field of proteomics, right? But if you think about the understanding the biology and looking at diseases, it's always important to look at the post-translational modification. Again, it was very difficult to look at various type of PTM analysis because if you think about from the entire global protein, what percentage gets phosphorylated, even less than 1%. So how to enrich those phosphopeptide and then try to analyze the proteome from that type of less abundant samples has been challenged. So sample enrichment using different type of IMAC columns and different type of titanium dioxide based technologies to enrich the phosphopeptides, followed by running them in the mass spectrometer with very sensitive instruments and acquiring all type of phosphorylated proteins or the phosphomap of the proteome is another thing which is emerging very heavily. And not only phospho for that matter, even glycosylations are very important and very complex and very challenging. And then think about biology for ubiquitination and acetylation and variety of type of degradation and many type of other type of modifications are also equally important to understand the biology and disease. So a lot of exciting things are happening in this area. Of course, in your theoretical course of proteomics and proteogenomics, we did not have time to touch upon all the latest advancements, but these are, kind of some of the quicker updates. And again, as I said, it was not possible without some of the very latest high resolution mass specs like from the Orbitrap, Astral Orbitrap, and with the timsTOF mass spec. Without them, it was not possible to go to that level of depth of these very important class of proteins, of membrane proteins and the PTMs, or looking at even the single cell proteome. But these are started getting more and more reality, which actually adds a lot of excitement for all of you who are learning these new areas and want to venture into this new field by taking up the exciting research problems in the future. So while these are some latest development, but what is the typical workflow of doing proteomics? For all of these, whatever I spoke, or in general, if you want to do quantitative proteomics, a typical workflow involves that you take some biological sample. It could be a cell line, a biological tissue sample, or various type of body fluid. It could be saliva, it could be serum, it could be cerebrospinal fluid, many type of biological fluid samples, right? All of them, you extract the protein, you digest them with proteases like trypsin, chymotrypsin, lysine, make them peptide form. And then those peptides, you are ionizing them with the electrospray ionization, ESI. After ionization, they are going to go inside the mass spectrometer, which could be a variety of mass specs. And then you are acquiring their M by Z, and then you are generating their MS spectra or MSMS spectra and going to analyze that data. Typical proteomics workflow, whatever type of thing I'm talking, they will fall in this kind of domain. Only thing which becomes very critical is the sample preparation part, which we already spoke in the last live session about different type of sample prep, how to make it more unique and enrich that kind of sample, right? But eventually, as you go along, there are different type of workflow being followed to look into the quantitative type of workflows or looking at a specific modification. And accordingly, you have to fine tune your workflows for the data acquisition at the mass spec level and data analysis level. So we have this high resolution mass spectrometry facility at IIT Bombay, which we have established as a national facility, which is equipped with variety of instruments. And what we thought, we are going to show you some of these instruments, how they can be utilized for different type of workflows. So for example, here you see Orbitrap fusion, which is really utilized for the deep proteome type of analysis or deep phosphoproteome analysis. Q-executive along with the HPLC configuration is heavily utilized for metabolomics, and triple quadrupole instrument is used for the targeted proteomics or metabolomics based analysis. So we will take more time in the lab and try to walk you through with these type of workflows. But to begin with, we will first talk to you about one of the quantitative proteomics based approach, which is is still pretty powerful, which is known as the TMT or tandem mass tag. So idea is, can you, you have 3 control sample and 3 treatment samples. You want to do quantitative analysis, quantitative proteomics. You digest the protein, and after that, after digestion, you have these peptides, and those N-terminal peptides being labeled with these isobaric tag, which is having different type of reporter ions. For example, tandem mass tag reporter ion 126, till TMT 131. These are all reporter ions. These are isobaric, although reporter ions are different, but then you add a carbonyl group to make them all same mass. And then you are mixing them with each of your 6 conditions. So now every condition of 3 control and 3 treatment, they all got unique barcoded, right? And now you can mix them. And after mixing them, you run in the mass spec. And then you can, when you look at MSMS level, you can now segregate these reporter ions separately and use this information of intensity to quantify the signal. This is one of the very powerful way of doing the quantitative proteomics. Other way of doing quantitative proteomics could be label-free quantification, where every sample you're not labeling, but you are going to run them individually in the mass spec, and then you are acquiring the data, which is more like you are building a library of every patient specific or sample specific, and then you are counting the number of spectra or the relative intensity of all these abundant peptides and trying to compare those across your control and disease condition to then come up with quantitative signal, how much fold change you can see from control and treatment. So 2 different ways of doing analysis. Both of these are coming as a part of data-dependent acquisition. There's also a new emerging field known as DIA, data-independent acquisition, which I'm not talking right now to keep it simple. But we will just walk you through about TMT workflow and how to acquire data in mass spec in a live session right now, which will be done with Dr. Argya Banerjee. He's going to show you the workflow and talk to you about the data, how to acquire and analyze the data. So Argya, please talk about TMT now.
So, admin, can you help me to share this screen? Okay. Okay, so let me minimize this. Okay, so welcome everyone. Welcome to the introduction to the proteomics course and TMT based proteomics. So first of all, like I will be giving like what are the advantages of sample multiplexing with the isobaric mass tags. Though professor has explained more of it, like how and where it is useful in the field of proteomics, how and where it is useful in the field of proteomics, it will give you increased throughput. When you are using isobaric tags due to the reporters and the I and yeah, just slide. Yeah. When we are using isobaric tags, there is a very few chance, like the missing values will be very few as compared to the label-free quantification. We have a vast range of sample flexibility, whether it is from human or bacteria or plant. Any species of the samples can be originated, and like it can help us in multiplexing, means comparing more than 2 samples. Means you can ideally now they have taken out a range of 24 and 26. Like in a one go, you can compare like when more than 2 samples at a time, and multiple comparison will help you to improve the statistics. Now, as the professor told, there is a mass reporter, there is a mass normalizer, and there is a protein reactive group. Your peptide will be connected with the balancer, and balancer is connected with the reporter. This is the reporter what you want to see. Coming up to the workflow, as you have gone through our earlier session where we have shown you how to prepare the samples. We generally prepare the samples. We do a fraction, we do a labeling of the TMT tags after the peptides have been generated, and are followed by a cleanup, followed by a fractionation, which can improve the coverage, followed by the concentration, data interpretation. Data analysis can be done with the free software such as MaxQuant and FlagPy, as also as the Proteome Discoverer, which is one of the proprietary software. Now, when you are dealing with the optimization of the TMT methods, 2 things you have to keep in mind. If you are using shorter gradients and shorter columns, that can increase the co-isolation. But if you are using 2 hours gradient with a 50 centimeter column, either you have to increase the length of the column or the length of the gradient. A 15 centimeter column, 1 hour to 1.5 hour of gradient is good enough. But if you are using 50 centimeter column, you can also load 500 nanogram to 1 microgram of load, which can be go to 2-hour gradient. So this is how the gradient establishment can be done. And as you know, like see, this is a C18 column, so more of the hydrophobic peptides will be there. So this is the interface, how the method addition is being done. So this is the method editor, which I am going to show you if the time permits. So here, like in the peptide quantification, you can easily go to the TMT MS3 and you can place it here. You can just drag this PMT and you can place it here. Once that has been done, you will be given an option like how you want to analyze your MS1 data and MS2 data. The MS1 can be done in the OT mode, and because OT have a higher resolution, and MS2 mode as this arbitrary fusion, what Professor Sanjeeva showed in our facility, is of tribrid mass spectrometer. Is one of the tribrid mass spectrometer. It has 3 kinds of mass analyzer: quadrupole, ion trap, as well as the orbiter. The second mass analyzer is a choice whether if you are using SCD, you can use OT mode like a orbiter mode, and if you are using CID, collision induced dissociation, you can use the ion trap as the mass analyzer. And thirdly, like in the DDMS2, which is the main for use for the quantification, you can use the OT. So in one, like you can go to the 12 lakh Dalton, like the 2 lakh Dalton of the orbitrap resolution, but you can go to in the mass in TMT mass specs, you can go to a range of 1 lakh 20,000 Dalton, and severely like a multiple charge state. The charge states can be concluded include from 2 to 6, because this is one of the robustness which separates from the multi-top, because at a single go you can monitor more than 2 charges: monoisotropic precursor, keep it to simple peptide, a full MSMS resolution with charge state, dynamic exclusion, and ion threshold. So these all are the settings. We are good to go, and consider selecting the most intense ion at a top speed mode. So yeah, so now this is done. When you come to the sensitivity and specificity, the isolation window is generally from 0.7 to 2 MOU. The choice can be between 5 to 10. And for best accuracy, top 10 matches of the pre-fractionated samples were taken. So this is how a ideal parameter or a ideal matrix of an TMT workflow lines. So this is a 2-hour gradient, that is 120 minutes. You generally start with less hydrophobic, means more of the aqueous, to high hydrophobic, which is an organic solvent, which is ACN, and you can easily obtain more than 2,000 proteins in a single shot, and 10,000 to 11,000 peptides IDs in a single go, and that can be quantified with the help of proteome discoverer. Okay, so now let me take you to the original mapping of the instrument. Just a So this is the original interface of the video. So that 120 hour minutes gradient, you can see it here. This is how a chromatogram looks like. And if I can show you the total ion chromatogram, view ion map. Okay, you can see the spectrum also. This is how the spectrum looks like. Most of the elution has happened from 30 minutes to 110 minutes. So this is our gradient. Like the first is the aqueous phase, which I have shown you 5% of B, then is the hydrophobic phase, and after that again aqueous phase. So if we zoom in, sorry. Okay, this is hanging. Software is hanging a bit. So if you zoom in, each peaks represent. So this is how the crux lies. So you can see each peak is a MS, and the void between the two is the MS. So this is how a peak is being seen. So if you click it here, let this also be there. You say view spectrum. Okay, great. So like you can see, so this is the MS. So once you shift, you can see if between two MS, if the fragmentation is of MS is more than 10, because we have selected top 10 abundant proteins. If the transition is coming more than 10, it is a very good spectrum. At indeed, it is a very good spectrum. So this is how you can monitor how the chromatogram looks like. Once this is done, you can see your labeling efficiency. TMT templex ions can vary from 126 to 132, whatever the reporter ion may be. So you can simply go here. You can go on the ranges. In the ranges, just a minute, let it come. In the ranges, you can select one more column where you can select MS. Sorry, we have to do it on the spectrum. So view chromatogram. Once the chromatogram comes, you can go on the. I have already done this. Okay. Go to the ranges. Once you go to the ranges, I will show you from before. Okay. So, so this is how it looks like. So this is a chromatogram view. You go to the ranges, and suppose my labels which are there are coming. I will click on submerge with another graph, and I will click on the mass range. Now it will give you the mass of the MS precursors which have been detected. So as we know, the labels which are occurring in the TMT tags varies from 126 to 130, 130 for 4 plates or 132, whatever may be the ranges. We will just click on the same. So this is how you can take out the mass range. You just click on OK. It will take some time. So it is going to tabulate and see what all precursor ions which are present here are labeled with that kind of labeling efficiency. It's taking a bit of time. And by the time it comes, like we can show the instrument. By the time this thing comes, we can show the instrument.
All right. So, well, these are the live run, which our guy is about trying to show you from the Orbitrap Fusion, which we are acquiring the data. And again, to really get, proper fragmentation here and looking at the reporter ions, you need to optimize the collision energy and you need to do a lot of optimization at the chromatography level and MSMS parameters. But eventually, if your labeling was highly efficient, you have done the proper labeling, and if you are able to now look at the data, you can acquire a lot of good qualitative accuracy. What you see now also on the directly from the interface, let's so Arge, if you can just unshare your screen, then we can have you on the full screen.
I'll see where the samples are injected. It is already loaded and it is running, so I cannot open the compartment box of the nano LC. The sample is eluted from here and it comes to the orifice. So this is the main easy spray source of fusion. And you can see like this is the trap column, what we are having. Two types of columns are used before the sample is injected into the instrument. One is the pre-column or the trap column. If any debris which is coming from the direct nano LC, it will be trapped here, and after that an analytical column, 15 centimeter analytical column is used in which the samples is injected. Sample is injected. So this is the ITC, isothermal calibration tube. Once the sample goes inside, it goes onto the instrument, and after that you can see that kind of gradient. So coming on.
All right. So thank you, Arghya. This is one of the workflow of doing the quantitative proteomics. And once you have identified, let's say, a lot of protein, which shows you significant protein lead, which is a good hit for you. So now you need to validate them, right? Conventionally, people used to validate the targets using the antibody-based approach like ELISA or Western blots. But for every protein, we don't have so nice antibodies available. For many, it is only polyclonal available. So then how to validate the target? So since we are acquiring data from mass spec, which is more about the peptide sequences and their quantification, idea came, why not we only validate those peptides from different type of mass spec. So we are, let's say, discovering an orbital fusion, but can we validate the targets of only specific peptides of the proteins of interest using the triple quadrupole based instrument or using another type of orbitrap instrument? So the two different workflows which are currently being heavily used, one is multiple reaction monitoring, other is the parallel reaction monitoring. Multiple reaction monitoring is one of the triple quadrupole based configuration when you are having the two set of quadrupoles, Q1 and Q3. In between, you have collision cell, and you already know that which of the specific protein, let's say 5 proteins of that 15 peptides of your interest, you have told the instrument with the hardware with a specific software on a Skyline that you're only going to look at the transitions of these 15 peptides, and only those the Q1 is actually acquiring data and doing fragmentation to generate the relative ion spectrum of that. PRM is much more powerful. It can actually look at all of those precursors and fragment ions simultaneously. So then one could go much higher number of transitions as compared to the MRM-based approach. So in which way these experiments can be done in the lab? Let's have another live demonstration from Avinash, who will walk you through these workflows quickly directly on the instrument. So Avinash, please show the MRM-based workflow.
So, hello all. So, as you can see, sir already discussed about the targeted proteomics. So, this is the TSQ voltage from the thermoscientific, and this has the basically the triple quadrupole. So, this has the two mass analyzers, and along with that, this is the LC system connected. So, this is the LC system, and every LC system has basically multiple compartments. So, this is the basically auto sampler where we keep our samples like in this kind of files. And so these are the different kind of tray where we keep our samples. And from these samples, they go to the one. This is basically the pump compartment, and this one is the yeah. So you can see these are the pump compartment where all the solvents have been mixed and get eluted during according to our gradient what we said. And this is the column compartment where we keep our compartment, we keep our column, and it maintains some internal temperature for the columns. And these are the solvents. Basically, from this solvent, the different gradient has been prepared. So, as you can see in the monitor, so this is basically the liquid chromatographic system application. So, this is the pump module where we can set our gradient, and this one is a sample, and we can set where is our sample is present, and this is the column compartment, and we can set the temperature. And all the method file which has been used for the targeted proteomics are set in the Tune application, and those method has been set in the X caliber. So as you can see, this is the live run going on. So these are the chromatogram and different kind of TIC and the gradient shown here. So as you can see, it depends on the what chromatogram, what gradient we set. According to that, they get eluted, and this kind of chromatogram comes. So I can also show. So this is the one of the sample run from the human sample. So this is not the Gaussian distribution as we say in the global proteomics. So this is the quite not that dense chromatogram, because this is we are looking for only the targeted proteins and peptides. So that's why chromatogram is not there. So I can show you some of the results from. So after that, getting the raw files for this kind of raw file, we can import those raw file in the different another software which is called as a Skyline, and we can look for the peak of a particular protein. So this is one of the study from the COVID. So as you can see, so upper top panel is basically the COVID positive, and lower panel is basically the COVID negative. So we have identified the multiple proteins which are basically shown the differential regulation in the different set of a group. So as you can see, also the intensity of in the negative sample and the positive sample. So basically, this instrument has been very good for doing the targeted proteomics and from the after analyzing the proteomics global data set. So yeah, over to you.
All right, thank you, Avinash. So now, let's say you already have looked at your specific protein targets, and you are able to now correlate that intensity what you have observed, the fold changes, they are same, whether you look for the discovery or now in the validation stage. Additionally, from the, since we're talking about mass spec is also very powerful LC-MS approach to look at the metabolites in a field known as metabolomics, right? The way we look at all the proteins in proteomics, likewise, mass spec is very powerful to look at all the metabolomics for all the metabolites. Although there are other techniques as well, like NMR can also be used, GCMS can also be used. They give you very specialized classes of metabolite, but a global broad survey can be done with the LC-MS based approach. So since let's say you think about you have biological sample or tissue or cell lysate from which you want to do proteomics, you're going to extract the protein out. Why not, you also extract the metabolite out on the same sample? For example, if you're doing methanol-based precipitation, you can even use the same sample for even some part for the protein, some part for the metabolite analysis, right? How powerful that could be if you are integrating the data from the same sample. Today's live session is also for the broader audience talking about proteogenomics and multiomics. So now we want to expand your view. Think about from the same sample you are generating next layer of information at the metabolite level. So metabolomics can be done using various types of mass spectrometer. One which we are going to show you is another Orbitrap Q executive, which is interface with the UHPLC. And it is pretty straightforward, much simpler to do metabolomics as compared to the proteomics. Of course, you need to optimize multiple parameters, which now Ankit is going to quickly demonstrate you metabolomics-based workflow directly from the lab.
Yeah. Hello, everyone. Good morning. So now what we would be showing you about the running of the sample for the metabolite identification. So as it was discussed that in the proteomics, there is the nano-LC or the nano flow of the solvent and the solution. But here in the for the metabolite, what we generally use is the HPLC. Here we use the flow rate. You can see it is in the range of ml per minute. So here is the same like in the voltage what Avinash has showed. There's the same compartment of auto sampler and the column. Now metabolites can be divided into 3 or more classes on the basis of their hydrophobicity. So it can be hydrophobic, it can be in between amphiphilic, it can be hydrophilic. So on that basis of that, the particular column is used. Now, if you are want to dilute out only the separate identify a hydrophobic molecule, so then a C18 column as it is being used in the case of proteomics. So then same reverse phase chromatography is being principle is being followed where the in the column compartment the column is being added, and in the solution you can see that it is being made the different layers, different gradient of hydrophobicity is being generated like you can see here, and on the basis of the hydrophobicity compounds are eluted out. Now after eluting out, as it goes into the mass spectrometer, and this mass spectrometer is a high-speed mass spectrometer where it is consists of the quadrupole and the Orbitrap. So there in these two mass analyzers, firstly the precursor ion is getting selected at MS1 level, and then finally at the after the fragmentation MS2 level is done. So I'll just quickly show you some runs that are going on. So you can see that these are the spectra at the MS level. So the metabolites, now one thing that needs to be understand that metabolites can be identified at MS1 level with the compound molecular weight, but same compound can have different molecular weight. So for this reason, we need to check the MS to match with the known compound. That particular compound will have a unique fragmentation spectra. So this is the run going on with the chromatogram on the basis of the gradient. The particular compounds are getting analyzed, and here if you see the runs that have occurred, so here you can see this is the particular compound that has eluted out. Now, in the case of metabolomics, we also need to add some kind of internal standards and run QC pulls to check that our instrument is not, there is no technical variability in between our runs. So, for example, this is one of the standards which which should be eluting out at any specific time throughout your experiment with a certain intensity and a good fixed shape. So, this is in brief about the metabolomics experiment, global metabolomics. Yeah, thank you.
All right. Thank you, Ankit. So again, some of these experiments, what you see, students who are live, it's not very difficult to actually perform these experiments, right? But what actually becomes difficult to really analyze this data, because it takes a while to make sense of these data. While it actually is very easy to generate these data, but to really get the maximum output from using these type of workflows of metabolomics or proteomics, you generate huge amount of data, and analysis and workflow that is kind of very challenging. I think, while we are going to talk more about other technology, let me just take a question from a student about TMT. How can we achieve high sensitivity in detecting low abundance protein using TMT-based proteomics? So in proteomics, is always a challenge that, whatever entire complex protein we want to analyze, there are always some abundant protein which will be having more peptides, and they will always mask our, mass spec ionization followed by MSMS analysis. So you always see high abundant protein much more as compared to low abundant protein. So it's actually, not the TMT or any quantitative strategy which can enrich out of low abundant protein, but rather your sample prep is the first place when you can think about how to enrich your complex proteome. Many samples, like, for example, you work on a plant sample, it has a plant leaf, has ribosco protein, very abundant protein. You work on the saliva protein, you work on the plasma protein. All of these samples, like, amylase or having the albumin, IgG, all of these kind of abundant proteins, you need to remove them to then try to enrich the getting the high abundant protein, and to be out and low abundant to be seen in the mass spec. So sample prep is the first step which can be utilized. Then once you are, let's say, done the extraction and done the your proper ways of enrichment for low abundant protein, the next thing is, can you fractionate those, so that you have from the one complex tube which has all the 10,000 protein, can you separate in 10 tubes, right? And with a different type of pH fraction, other type of chromatographic method or isoelectric point method, and fractionate those. Then each one of those, when you are going to run in the mass spec, you are going to see abundant proteins, which are low even abundant, will be enriched. Now, TMT will be now very powerful because TMT is very accurate for the quantification. So if you have done these kind of pre-sample processing and the fractionation, then TMT-based approaches will be able to, do more accurate quantification of the low abundant protein, which you were not able to do earlier. But your sample preparation strategies and fractionation strategies will make a very important role for giving you the broad coverage. So we'll be happy to take more questions. So please feel free to ask on the forum if you have more query. But let's just take, kind of to broaden the scope. We are talking about multiomics. I just talked about proteomics and metabolomics. Think about genomics, why that is the most important, like, the blueprint, right? So can we even try to think about integrating the data from genome and proteome, right? And that sometimes, especially for disease context, can be very important. Genomics is very powerful for sure and very robust. It can, very quickly with the next generation sequencing can look at all possible, your gene sequences, the various variant forms, looking at mutations, all type of things. And if you were to look at from the same sample, the protein, and the same sample, the metabolite, you're building the layer by layer information. Now, from the genome level, you are able to see, okay, which are all possible mutation from that given cancer or given disease, you are able to see. And now using protein, you are able to further enrich and say, okay, how many modifications I can see? What is the correlation between these type of genotypic changes which we see and finally happening at the phenotypic level by integrating the functional information from the proteome and the metabolome? So this is where I think integrating the piece of information is very important. And you are taking this course on proteogenomics, you have gone through it, and there is this, very active effort with the proteogenomics community to look at the, especially for the cancer program, which was started by Joe Biden about cancer moonshot, to look at integrating the data of different cancer type and provide much more powerful information as a part of a big initiative known as the ICPC, International Cancer Proteogenome Consortium, where India is also now part of the Cancer Moonshot program. So Aishi is going to show you briefly in the lab about one of the genome sequencer and some brief overview about it. Then we will shift to some of the big data analysis part.
Hello everyone, I'm Ayushi and I'll be giving you a demo on the NGS instrument that we have. So currently we have Ion Torrent S5 instrument that you can see in your screen. I'll give a brief overview on the technology and then we can talk about the instrumentation. So if Ankit can focus on the screen. Yeah. So I'm going to talk about the Ion Torrent S5. So a brief overview on how you can prepare your sample for the genomics. So first step is to prepare the library. And to prepare the library, very initially, you should have a good genomic DNA or any kind of DNA that you want to do for the sequencing. You need to have that DNA, good quantity, and you quantify that DNA followed by the amplification. Once the amplification of the target is done, you have to digest the amplicons. Digest the amplicon in such a way that the sticky ends are. Yes, sir.
I think probably your screen is shared. You have to stop the sharing of your screen. Then we can see you in large, because right now it's very small panel to really view.
Okay.
From your system, probably have you shared a screen something that you have to stop?
Sir, I have not shared anything.
And this frame of Ankit has to come on the full screen because you are very small, minutely seen.
Okay.
Ideally should come on the full screen, right? Of your part. Then only it will be visible. Now I think it's coming.
Okay.
No, you have to. Okay. Its PPT is coming actually.
Yeah. So first I'll explain the PPT and then we can give a demo of the instrument.
All right.
Yeah. So as I was saying, the first step in the next generation sequencing is to get a good quantity of the DNA. And once you have a good quantity of the DNA, next you can amplify the targets. There are multiple techniques by which targets can be amplified. One of the most common method is the PCR. And once the targets are amplified, then the sticky ends are created. As you can see here in the workflow, the targets are amplified. You will get multiple copies of your target DNA. And then the sticky ends can be created using mild detergent. Once those sticky ends are created, then the adapters, barcodes, which are essential for the sequencing reaction, will be added to those sticky ends. And now these are your libraries which are prepared. The next step is to amplify those target libraries. For that, in our Ion Torrent S5, we have an instrument called Ion One Touch 2 instrument, which works on the principle of emulsion PCR. So this is how the instrument looks like, and you have to fill the emulsion mixture here on this setup. And once it's there, the emulsion reaction will be started, and the libraries that you have prepared during your first step of NGS will be amplified. Followed by the next step would be to enrich those template specific target libraries. For that, we have another instrument which is called One Touch ES. This is how the instrument looks like. So you will just enrich your template specific target libraries in this step. Once the template specific target libraries are enriched, in this step you will remove those beads which are not having any kind of library. So those libraries, those beads will be removed in this step, and you will have proper proper template specific libraries, and those libraries will be loaded onto this chip like this, as it is shown here. Using 1 ml pipette, the libraries, template specific libraries, will be loaded on the chip. I'll be giving a demo of this chip and everything in a few minutes. And that's how you can, once you have your template specific libraries are loaded, you can just load this chip and start the sequencing run. Talking about the principle, how this Ion Torrent S5 works, so it works on identifying this proton ion, which is released during the formation of phosphodiester bond. So we all have learned molecular biology, where we have learned that during the formation of phosphodiester bond, whenever one nucleotide is added to the ongoing strand of DNA, there is formation of phosphodiester bond, which is something like this, and there is a release of proton ion. So this instrument basically detects this proton ion, which is released during this step, and it converts this chemical energy to the electrical energy, and that's how. So this chip has a lot of sensors here, where this proton ion is detected by the sensor which is located below the chip, and it converts the chemical signal to the electrical signal. That's how the instrument gets to know that the sequencing is happening. Now, if Ankit can show the instrument, I'll give you the tour of the instrument. Right. So if you can focus on here. So this is the ion torrent S5 instrument where the sequencing happens. So before to start the sequencing reaction, you have to clean the instrument. So I'm going to perform that step and I'll show you what all reagents and what all cartridges goes inside for your sequencing. So it will take some like just few time. So you can see here, it will tell you that these are all of the cartridges and all of the wash reagents you should have before starting the cleaning of the instrument, and then subsequently the sequencing run. I'll show you. That's how the inside of the instrument looks like. अरे यहाँ से ये कैसे दिखाएगा? So it's stop. Okay. Wait. Sir, I'm not even there. I have left the meeting from here.
All right. So she is figuring out, a way to expand her screen. What is important here to think about the different technologies available, whether for looking at your complex proteome or genome or metabolome, but can we really be creative to look at from the same sample and try to enrich that data, right? And how to really look at the entire thing in more totality. So now I think you can see the ion torrent instrument, and I should briefly please explain. Timing is less now. Aishi, go ahead. Can you?
Okay, so sorry for the glitch, but now I hope you can see the instrument here. So this is the ion torrent S5 setup that we have in our Proteomics lab. And before going for any sequencing run, we have to first clean the instrument. So I'm going to show you how to clean the instrument and what all reagents, cartridges go inside the sequencer. So Avinash, you can focus on the instrument now. That's how the instrument looks like. And I can show you inside the instrument where we have all these cartridges and everything. I'll just this chip clamp where we have this chip, which is very important. It looks something like this, and here are a lot of biosensors which I was talking about. And you have to load your sequencing reaction onto here. And once your sequencing reaction is loaded, it should be equally spread along this chip. And then we can gently place this chip onto this chip cartridge like this. It will go smoothly, and you have to close this chip clamp. Other than chip, this is the cartridge, which is the nucleotide cartridge. Here, all the DNTPs are here, and it will go like this here, like this in the instrument. This is the wash solution. So every time once your nucleotide is goes to the instrument, this is the wash solution. So every time after the one nucleotide, the wash solution will clean the tubes and everything. This is the cleaning solution, which is going to be used during the cleaning. And here we can also spot the, this is the waste collecting thing, where all the waste of the sequencing reaction will go here, and you have to clean this cartridge after the sequencing reaction is complete. So that's how you have to be very cautious while checking all the things are in place. And once
All right.
that is done, you close the
If sorry to
instrument.
stop you. I think we are getting late. Thank you for this demonstration. Yeah, so broadly now you can see a lot of data can be generated, but how to analyze the data? So timing is a bit short, but we will give you at least some flavor of looking at first the mass spec data. And mass spec data analysis can be very complicated, and the software which are currently available, they can take you like good RAM of your mass spec, and it can take few days time. But a new software which is developed from an academic lab, Dr. Alexei's lab in Nashville, in US, MS Fragger. It is one of the very highly recommended software for you guys to try out. Even in your own laptop, you can run some complex mass spec data and analyze that. So Avinash is going to give you very quickly just the kind of software interface because we don't have time to run the data right now. But again, if you have really interest, you can always ask us. We'll be happy to have a separate session for you for the data analysis part. But Avinash, please quickly give a demonstration of that software.
Yeah. So, sir, can you unshare your screen? Yeah. Thank you. So this is sir, I already mentioned that. So this is the first after opening the software. So this is the FracPipe 22 version. So you can easily download this software by using going in the Google and just type the FracPipe. You will easily get the all the tutorials and the downloading links. So after downloading the links, you will get this kind of interface. And this here we have to install the multiple plugins for this software. So like MS Fragger, Iron Content, Die Tracer for the different kind of analysis, and the Diane and the Pythons and also the for the different software for the analysis. So now we will go for the workflow, like how we do the analysis. So after downloading all the these sessions, you can just click on the download and you can download all the sessions. After downloading, you can go to the workflow, and you can for this analysis, I'm just showing for the DDA analysis. And so this is the list of the different kind of workflow has been given for the all kind of analysis like DDA, DIA, TMT, and you can get all kind of data sets. So currently I'm using the LFQ-MBR for the DDA files. So just click on the LFQ-MBR and you can click on the load workflow, and it will get already loaded and will load all the parameters. So after that, you can add the few files. So I've already added here. So you can just click on this MS data type. You can just click on add file, and you can download this. You can just select all the files and you can upload here. So after that, after downloading the file, it will by default take the data type. If it is not taking the data type by default, you can just click on that and you can change the data type. And also you can also add the different biological group. So in this case, we have taken the 3 control and the 3 tumor samples. So likewise, I have annotated. So you can just click on the those samples, and you can change by using the conjugative way and by file name, and also you can do by the custom way. So there are multiple way of doing these things. So you can do this part in the same way. You can if you have the bioreplicate and technical replicate, you can mention this part. So this is the first part of loading the database, and after that you can directly go to the database part. And if you want to download or you can upload, so there are multiple way to upload your database. So some of the few database has been already given for the human muscular sensor and different other species, and also it was give the checkbox for the review sequence, add decoy, and the add common contaminant. So for this study, this is the human cell line. So I'm just using this by default one, and I can just I will just click on the OK. So after that, it will be get downloaded. And suppose if you want to add your own database, you can just browse and you can add your database here. And after that, the next part is basically all the parameter, the major parameter we set for the analysis. So you have already selected the LFQ-MBR workflow. But if you want to change the few parameters like missing cleavage and the enzyme type, you can change this part. So this is the showing the precursor mass tolerance and the fragment mass tolerance. So by default, it gives the 20. If you want, you can change according to your criteria. So and also you can also add what kind of diation has been done. So there are different kind of drop down has been given. You can change your enzymes and the missing cleavage part. And this section basically you can add your modifications. So by default, they add three modification, also two variable modification and one fixed modification. So one is the oxidation of methionine and the N-terminal acetylation, and the one another is a fixed modification.
That to be quick.
Yeah. So after that, you can just directly go to the quant MS1. You can check, just click on this part, and if you want to change anything, you can change here, or otherwise you can just directly go to the run and go to the browse and select the directory where you want to save the file, and you can just start, and it will just click on the run. So it will start running this way, and after the finishing this job, it will show this kind of interface. So your job is done, and after running this file, you will get the output like this. So this will be output file, and you will get the combined protein, peptide, and annotation file from this data sets. Now we will move for the, like we are more rushing for the time, so we will move to the further analysis. So you can currently they have added the downstream analysis software as well, which is the track pipe analysis. If you just click on this, it will directly open up. So I've already opened up this software, so the interface will be look like this. So yeah, so this will be interface for the fact pipe analyst. So you can just click on the analysis. You can select your data type. So our data type is LFQ. So we will select that, and if you can just browse the whatever the file was given. So you have to select the combined protein folder. Yeah, combined protein. So it will already get uploaded. After that, you have to give the annotation file. So this is our experimental annotation. And after that, so yes, it is get downloaded. Here you can select the max LFQ intensity, and if you want to change like various other parameters like missing values and the p-value criteria and the fold change criteria and type of normalization, so you can check on that. And also there are different type of imputation method has been given. So persist type, KNN. So those are the basically commonly used methods, and also the Benjamin Hodgberg for the FDA correction. And you just click on the run. So yeah, it will just take the few seconds to complete this part, and it will show like all the QC parts. So yeah, as you can see, so the first part to check the so these are the basically QC plot to check the data quality. So you can look for the PCA plot, sample correlation, sample series, and the density plot, and the missing value heat map. And you can also, overall you can check all the type of QC plot from this data sets. And after that, you can get the volcano plot, and so as you can see that in this it is showing a 4, 5. So we have not added the missing value criteria. So it will give you the list. So you can just sort on the basis of the like fold change criteria and like what fold change is coming. So it is also giving the volcano plot, and you can download all the plots and the tables which has been given. You can also add like what kind of enrichment you want. So, and also it is, if you want, you can look for the pathway analysis, this part. So this is a very convenient pathway, like convenient for the downstream analysis, and we can get the DPs from this. And after that, we will go for the pathway analysis, which Ankit will talk about that. So that's all for this side.
All right, great. So thank you, Avinash. I think it was a quick demo. But what users and the student who are joining today, they can see that even looking at, please stop sharing the screen.
Yeah, yes, sir.
Even now analyzing the complex mass spec data is very much possible. And earlier, we used to then look at different software to do the plotting of the data, especially the MS-Frag, like, MetaBoneList and Perseus, many other software. But now I think, you have advantage that you can use the MS-Fragger and generate from the same software, you can look at very simple output as well, and very fast. Additionally, you can always even do more of the proteogenomics data analysis with the cBioPortal and WebGistOr. You've already learned some of these tools in the proteogenomics course if you're doing that. But students of the proteomics, you can know that there's a world beyond just proteomics when you can now start thinking about integrating data and make it more multiomics, kind of picture. And during the short time, we will be skipping the demo of other proteogenomics tool. But what you have to really understand, what we really want to emphasize, that now there is a field which is known as multiomics or integrated omics, which is now driving the systems-based approach of understanding any complex system much more comprehensively. That's the field which is going to really make the difference, because in our actual physiology, whether it's human or other kind of living system, we don't have the gene, protein, RNA, metabolite. They don't work separately or independently. They all work kind of like, in a very relay race where every partner is very important, right? So when we look at only one side of the picture, only genome or only transcriptome or only proteome, we miss out the complete picture. Therefore, what is now much more with the robust technology and the instruments available, what is now much more possible to look at different layer of information from the same type of sample. And that's really more important for the complex disease, like, for various type of human cancer, diabetes, heart diseases. Now people are trying to look at layer by layer information. And then now what you realize is that since you're generating so much amount of data, it can't be only biology project. Now we are talking about the big data. So therefore, people from the statistical background becomes very important to look at the significance of the data. People from the core computational background becomes important to look at the entire programming, and even from the chemical background to look at the simulation of the data and come up with the much more systems modeling to understand a given system much more comprehensively. So all this is really, moving in the direction of what we see, like, the new era with the artificial intelligence, machine learning, to really make much more predictions. But predictions will only come if we have the robust set of the data to build the models. So therefore, right now, this kind of omics field is really playing an important role to generate very meaningful information and data sets, which can be utilized to make very important hypotheses, which can be further tested with the specific type of experiments. I hope you got the kind of a gist of various technology, which you have been studying in bits and pieces in the course, but at least you had the first-hand opportunity today from the lab to see some of these instruments and the workflows. So with this, let me thank all the TAs who have really given you a nice demonstration in a very limited time what we had. And also they have been following upon all of your queries on the forum and giving you, some meaningful comments on your various type of queries. So thanks to the entire team. And of course, thanks to all of you, the students who are really showing good interest into the course, are asking some very interesting questions to us and a lot of query which comes to us. And hopefully, like, some of you might be liking this field to utilize it for your own research or to take up the future job opportunities and career opportunities. Many students ask us this question, like what kind of future opportunities we may have in this area. And of course, given the significance of this field, you will expect that, okay, you are going to get a lot of job opportunities in this area, because people who can analyze these type of data sets will be in big demand. With this, I think we will close today's live session, which was our second live session for this MOOC course. And we really wish all of you the best for your writing exams. If you do well in your this MOOC course, you will have opportunity to come to IIT Bombay, spend some time in our lab, or do some internship or attend some workshop. So the journey does not end here with this kind of, the video courses, but rather we want to have to see some of you more live in the lab. And just, on the lighter note, there has been several MOOC toppers who have really, eventually joined our lab. And I have been really, very proud to see their career progression and our interaction with them. That they were virtual students who, then performed very well. They came to join our lab as a project student or interns. Eventually, they got into the PhD programs or MTech programs. They finished the programs here, and now they're doing very well for their postdoc and jobs in US and other places. So it has been really, remarkable, positive journey and experience to see how the virtual interaction from all over India and different parts of the world brings people together, and then we are helping them to move forward in their career trajectory. So we wish all of you the best, and will be very much available to take any other query and your other kind of future whatever apprehensions you may have and questions you may have, to give you much more meaningful insights of your query. With this, let me thank the entire team and close today's session. Okay, bye everybody.